Um, Hi, everyone. This is Let's Discuss paper soon after reading it. We selected and started reading a paper 2 hours ago. And from now on, we are going to discuss the paper. And today we have two people. Ryoichi, could you introduce yourself?
Okay, hi, I'm Ryoichi Watanabe at Kyoto University, Japan, and I'm a PhD student.
Thank you, Ryoichi. And me, Yota Kawashima. I'm a research assistant at ATR and Monash University, Australia. And today we are going to discuss the paper titled Behavioral Paradigm for Measuring Perceptual Distances in Mice, published in Cell Report Method, I think in this year, 2022, by Hirofumi Nakayama, Richard Gerkin, and Dimitri Rimbergs. Before starting the discussion, I'll just quickly give you a quick summary of this paper. And okay, so in order to understand neuromechanism or sensory mechanism, it's very important to understand the sensory perceptual space of that. And that requires some kind of distance measure between perception or stimuli. And this will reveal the structure of the perceptual space. And in human and non-human primates, there are several research measuring similarity between two stimuli. But in other species, such as mice, such kind of studies are really done. But, as you know, the mice and other animals are very, useful in the sense that we can use very high quality or high resolution monitoring. And also we can do some brain manipulation, such as optogenetics and things like that. So developing or establishing behavioral paradigm to measure distance between two stimuli is really important, especially if you can do that for non-primate animal, it would be great. So these studies focus on mice and develop behavioral method for that. And specifically, this paper, the authors focused on odor stimuli for a couple of reasons. First, it's easy to train mice, and also it's less explored the odor is less explored. So that's the reason why the author picked the odor instead of like vision or audition, things like that. So the aim of this study is to establish high throughput measurement for mice odor perceptions. So the purpose is to get the similarity measure between odor perception from mice. And in this paper, they proposed a method utilizing delayed match-to sample paradigm. And this paradigm, the mice were trained to respond differently depending on the similarity of the two sequentially presented odors. So if the two identical, the same odor stimuli were presented sequentially to the mouse, mice were trained to go to the center water spout, so that they can get the reward. On the other hand, if the different odor stimuli were presented sequentially, then mice were trained to go to another side water spout to get the reward. So depending on the identity or the similarity between the two odor stimuli, mice behave differently. And so here, because of the training, if the stimuli were similar, then mice will more likely to go to the center spout instead of the side. So by measuring the response or the focusing on the probability of response going to the center versus side spout, we can measure the similarity of the two stimuli from the mice. Then in this paper, they check the validity of this paradigm by first checking the whether the metric, the similarity measure, satisfies several mathematical conditions, such as identity, similarity, triangular inequality, non-negativity. And also they measures whether the, and also they check something like if the, let's say, if the mixture of two orders result in, for example, like if the order consists of two components and if they are shared between two stimuli, then the similarity response will be, or the distance will be less than the pairs of stimuli which are not, do not share the components. And also they focus on the similarity of chemical compound, and they confirm that then the chemically similar stimuli tend to result in similar perceptions and things like that. And also they did some modeling regression analysis to confirm that the probability of the response, the rate, whether going to the center versus the side spout, is predicted or is based on the perceptual distance, the similarity, instead of the, let's say, sequence of the stimulus presentation or the concentration of other things like that. And also they check the across mice consistency and found out that the similarity is consistent across mice. Yeah, that's my summary. So this paper basically proposed a method to measure similarity between two other stimuli from mice. And yeah, that's my summary. And Yoichi, do you want to add anything?
Yes, I totally agree with your summary. And in this study shows that the differentiation between the acid and non-acid is more different from more different performance is high than the within acid and acid one, right?
Yeah, so I think you're talking about one confirmation approach, and here the assumption is that if the two other stimuli is similar in terms of chemical compound, say if two stimuli contain the acid, then that should result in similar perceptions. And then they confirm that by comparing the acid order versus non-acid order, and seems like their assumption or prediction hold for their similarity measurement.
Yeah, I see. Okay.
Okay, if nothing, we can start discussion from now on. And do you have any comments, question?
So I'm wondering, yes, in this study, the author used the proportion of the rate proportion of the no-go task as our similarity judgment performance, right? But I think it is just the performance rate, not the similarity rate. So firstly, this point I'm now wondering, it is, yeah, so I think yes, discrimination is one of the measure for the similarity. I understand, but it is not just direct. It is not directed measurement for similarity judgment. So, but yeah. So I'm wondering. I'm I'm wondering this point.
Yeah, I mean, I totally agree with you, but you need to think about, let's say, if you do human, study, you can it is very easy to ask the similarity because you can just present two stimuli and then say, oh, are they similar or how similar they are? We can do that. So we can get the, similarity rating per trial. And this is nothing to do with performance, of course. But mice case and those other animals, like even primate, what we can do is just measure the performance, and based on that we can infer the similarity of their perception of two different stimuli. So yeah, but yeah, of course, yeah, they measure the performance in a sense. Yes.
So
yeah.
And so and we can add the measure for the reaction time or something. So metacognitive measurement, right? If the two stimulus is more different for mice can easily to detect their difference. So the reaction time is less, less. But if mice can discriminate two stimulus, it's different, but more similar than that one. So if so, the similarity is less than the other one. So the reaction time or something, the other metacognitive measure is more, how to say, if the reaction time is larger, if the two stimulus is similar. So
yeah,
I think
that's a good point because we can definitely measure reaction time, and then as you said or as you explained, we can predict that if the two stimuli are similar, mice will kind of hesitate to make response.
Yeah.
The response time will be longer for similar stimuli than for completely distinct stimuli. So yeah, that's a good one possible measurement, I guess. Yeah, that's a good point. And yes, and do you have anything?
And also I have another one point.
Yeah.
So why are the symmetry data is plotted the three-dimensional one? So what I want to ask that the yes dimensional information is how to be decided. Why the authors use three-dimension, not two-dimension?
Okay, I think I can answer that question based on my experience, data analysis approach. I think what they use is, first what we get is this, distance matrix, right? And there is some algorithm that we can, plot dot within a high dimensional space based on that, distance matrix. So it should be, basically the high dimensional. It can be even like a higher than 3-dimension, 4-dimension things. Then what we do basically is just, extract or focus on only the first 2 dimension that explain that data well. And yeah, as you said, we can of course plot the two-dimensional plot, but this, I think this also decided to use the three-dimension because if you only plot two-dimension, then we can't see the the third axis, which I think in this figure captures something. Let's say the order will be vary along this third dimension. So that's just how it works, I think. So my point is that 2D is not the, or usually this kind of algorithm provide a high dimensional space instead of two.
Yeah.
So, yeah, that's my answer.
I see. But how about 4 or more higher order dimension? It is so difficult for understanding us.
Yeah,
or 3D.
Yeah, first of all, it's very difficult to plot the 4-dimension, right? Yeah. That's one reason. And another reason could be that even if you include the 4th dimension, that will not explain the data well. So there are no necessity to, plot the data in the 4th dimension. For example, if you can, explain the data well only within 2D, then we don't need to add the 3rd dimensions.
I see.
But here, yeah, that part I'm not sure. Maybe, the reason why they plot 3-dimension here is only the difficulty of the presentation or visualization, or either, I don't know, just the 4th dimension do not explain or the captured data well. But that's my guess. Yeah.
Okay, I see. Thank you. So I have the other one, but after you.
Yeah, okay, sure. So this paper, in order to validate their method, they checked whether their metric, satisfy the mathematical conditions. Yeah, again, I said that identity, non-negativity, triangle inequality, and similarity. For similarity rating, yes, I agree that we should, or the metric should satisfy identity. Because if we compare the exact same, stimuli, then it should provide 0 distance. And non-negativity as well, because I don't know how to explain the -1 distance. Or of course, if you say distance, negative does not exist or should not exist. So that part I understand. But for the rest two, triangle inequality and similarity, specifically the similarity, as you know, or as our lab or now's project is something to do with asymmetry of similarity, right? So asking a similarity between A and B is different from the distance, or the not distance, the similarity between B and A.
Yeah.
And I think, or in this paper, they said that, for some stimuli, for some orders, this symmetry does not hold, what they said. So it's kind of support that, this similarity is not symmetric. So should not be, distance. And that part, was interesting. And also, as I mentioned, this paper is quite relevant to NAO's or Korea structure project. And, this paper shows that we can apply the, the similar method. And also mentioned that advantage of using, mouse models. Of course, in this paper, they did not show any data that, electrophysiological data. And did not do any, optogenetics, because the purpose of this paper is to provide or propose the behavioral method. But I'm really interested in or how we can do, or how we can, the future research based on this, method. That's really interesting.
Yeah, I see.
Yeah, my question, that's all my comments. But my question is that, okay, so do you think we can apply the similar method to, let's say, infant babies, which is your interest of research, right?
Yeah, I see. And this method, how to say, I forget, matching sample method is more famous or popular in the animal study, not only animal study, but also the infant study. For the like the looking paradigm, for this, if the same children's looking pattern is transferable, but if the stimulus is different to infant, more looking more time than the another one. So these methods, so the matching sample method is more fitted to the infant study. Yeah, as you said, but this sequential one, the one stimulus and then the second stimulus happen, and this stimulus is similar or not. It's a little bit difficult for infants because it needs to train the response for the stimulus. Like in this study, mouse trained before the test one stimuli and then two second stimuli, and if similar phase is on, that not similar phase didn't on, so off the phase. But this training is difficult for infants, I think. So just at the same time, the two stimuli is presented, and then how to looking is the best way for infants. But I don't know sequential method. Yeah,
I see.
Sorry, but we can, so how to say 循化 in Japanese, but yeah, your more habituation method is similar to this one. Like a pattern is stimulus is presented many times, then infants habituate this stimulus, and if the same stimulus is presented, infant is habituated this stimulus, so infant looking time is less. But if the other stimulus is it's presented, infant think or recognize, oh, this stimulus is different from the last stimulus, so infant looking time is longer. So this paradigm is, I think, related to this paradigm is better to see the sequential similarity judgment, right? I see.
Yeah, regarding the habituation task, I think in the introduction, and the author mentioned that there is already proposed method utilizing the habituation to get similarity from mice. But the problem is that you need a lot of time.
Yeah.
So in a sense, with this method, they said that we can resolve that. And this is the high throughput measurement, and that's the advantage. But I understand that part. It's kind of difficult to, train infant to make response for these tasks. But,
Yeah.
I see.
But I think in this study, the authors used the how to say order stimuli, so they cannot present these stimuli simultaneously, right? So order information is conjuncted to order. So first and second, the order stimulus is presented, right?
Okay. And now I think it kind of makes sense again to, it kind of makes sense why the similarity does not hold for this. I don't know about the order or auditory, stimuli, but it's easily, I can intuitively think that if the first, order cause some elicit the chemical, reaction at the nose.
Yeah.
And then there might be some adaptation or something happening. And then that will change the response for the second order. I don't know.
Yeah.
It might be, depend on the duration of the presentation. But, having said that, there should be something happening and the order is important.
Yeah.
So, yeah, that makes sense. And yeah, as you mentioned, using order as a stimulus is a little bit difficult because we can't, present it simultaneously.
Yeah, so yes. And in this study, the duration between the first one and the second one is like 5 seconds. I don't know the 5 seconds is enough for this chemical processing or the perceptual differentiation for the time or for the perceptual discrimination or chemical discrimination or chemical habituation. I don't know, but I think 5 seconds is for me so long to if the visual stimulus if we use the visual stimulus 5 seconds is so long. But in this study the study for the auditory of smell stimuli, so the five some compared visual or audio study, the more long duration of the two stimuli, I think.
Yeah, I didn't pay attention that in the stimulus duration, but as you mentioned, 5 minutes a little bit long. So maybe in the supplementary or the method part, they mentioned that they want to avoid the effect of the first stimulus on the second stimulus, and for that they use this 5 second duration. Yeah, but okay, maybe that might be the case. But yeah, okay. So any other and other comments I would want to make is that this across mice consistency, and again this kind of related to Now's proposed massive report paradigm, right?
Yeah.
Because if we can confirm that the response will be or the perception will be similar or consistent across mice, then by grouping the mice into some set and then presented different set of stimuli, the order stimuli to mice, we can get the huge amount of responses, or the pair, we can present the huge amount of pairs of responses, so we can get the rich structure of this all the
Yeah.
space. So that part, yeah, I think this, is highly related to now's, massive report paradigm.
Yeah, I see. So there are the most researchers in the world. So we can do the massive study of the each lab's mouse and mouse similarity judgment data and integrate it. And yeah, so it's so interesting.
Yeah, and also like if we can do that for mice, we can do the same thing for adult human at least.
Yeah,
and then we can get the perceptual space from human and also mice, so we can check the consistency between species. And if the shape is similar, then we can say that how we feel the order is something similar to mouse, but maybe it's quite different. And also here the paper focus on mouse, but I guess we can apply similar approach for other animals, like I don't know, dog or other primate, monkey, and then we can get structure from different species, and then we can compare.
Yeah, I see.
Yeah, that's really interesting.
Yes, and I think this is the next step of the Korea structures, like the how to say
エボリューション
evolutionary Korea structure, or for me, and the developmental Korea structure, right? So interesting.
Yeah, but that's my comments. And this again, this paper was really interesting.
Yeah, interesting. And I have a one comment and a little bit question. So, how about the similarity circle? It means that if we think the color similarity, we can measure the physical color information, right? Red and like the RGB, red, green, blue are factors information. So objective information can measured, and we can compare the subjective judgment similarity rating and objective color information. But I don't know I don't know that this how to say smell or objective smell information or objective chemical information. So objectively, these chemical is similar or not. I don't know. So then, so subjectively similarity of this chemical information in rat, mouse, and also it is important to the objective similarity in the chemical one. So
I want to say one thing, that is, I understand why you say subjective versus objective, but for me, both, this paradigm use the objective measure instead of subjective measure. But I understand what you want to mention. But that part, at least they confirm that, or they check the acid versus non-acid. That's what you say. That's what you mean when you say objective, right? Like the structure of molecule or something.
Yes.
Properties. Yeah, that part, maybe we can or they can do something, but I don't know. Yeah, because I forgot almost everything about chemistry. So intuitively, we can say that if the structure of molecule is similar, then they will have similar properties. So they will result in similar, other perceptions. But there, many aspects we need to consider, right? Like what constitute that molecule, like, I don't know, carbon or oxygen or things like that. And yeah, I don't know whether that's interesting.
Yeah, but possibly the color is special, like the closed circle. I mean, the similarity or color circle is right, red, like the circle. I assumed that the color information is like the objective circle. So I think I have this question, but normally in the world there's not the circle relationship. The more wider or more open relationship in the of the like the stimulation or factors in the world, maybe.
Okay, so now I get what you want to say. So I think what you're saying that for order, we can't make any prediction on the structure of this, similarity, right? So if we do something similar on vision or color, you said we can predict this circular relation, the red, blue. But for this odor, the smell, yeah, it's true that, what kind of structure we expect. But I guess, I guess first we do not have any prediction. Then do this, experiment for many other stimulants, and then we get some, gigantic structure of order, right? And then from there, maybe we can say that, because we have this structure, we should be able to see something similar for neural activities, can be related to anatomy, or maybe the, I don't know, receptor of, nose or something, this for smell. Yeah, but yeah, I understand your point. Yeah, I don't know, like, when I see this figure, the 3D structure, it looks fancy, but yeah, it's a bit difficult to, say anything.
Yeah. I see.
Yes. Okay, yeah, I get your point. Was that your last question, comment?
Yes.
Okay, so if so, we can wrap up here and see you next week. Bye-bye.
See you.